With the new release of Mastodon Deep Lineage, we also had the possibility to perform dimensionality reduction using the Umap algorithm. The motivation behind dimensionality reduction methods, such as Umap, is that you can analyze multiple data dimensions at once, for example, in 2D space. Built into Mastodon, you have this compute features functions, and that compute features functions can compute for you quite a lot of features. So for example, the spot intensity, which is a feature that is based on the actual image data. You have the spot ellipsoid aspect ratios, and you have some more spot ellipsoid properties such as the volume. It's also possible to compute the speed, and on the branch level, so the branch describes the cell between two cell divisions, so from its birth until it divides into new cells. There are further features available such as cell division frequency, branch movement direction, branch sinuosity, branch duration and displacement. If you would like to know more about these features and how they are defined, I would like to refer you to the Mastodon documentation. With the grapher that comes with Mastodon, it's possible to plot these features in XY space. So for example, you can plot the mean intensity of a spot in channel 1 against, for example, its volume. And there you can study like the relationship of the intensity to the volume. So here you can see that the brighter cells are usually the smaller cells, and also only in the darker cells you have like a distribution. You have small and big cells. Another example would be if you plot, for example, the intensity against, let's say, the velocity. And there you can study this relationship. So you can see that both, so the dark cells and also the bright cells, that most of them, they move really slow. And there are only a few cells that move a bit quicker. Okay, now what happens if you would like to study more than 2 dimensions at once? This is with the grapher not possible, but could maybe think about the 3D grapher that shows data in 3 dimensions, but yeah, more than 3 dimensions is difficult to display. And for this purpose, there are algorithms that allow dimensionality reduction of data. And this is now built in. So you can see here in the table, there's lots of data columns that can be computed. You can only choose 2 of them to view in the grapher. And in order to overcome this situation, it's possible to reduce those multidimensional data to 2-dimensional data. And the purpose of this might be that you want to identify patterns in cells, maybe even cell types. And to do so, it's recommended to mainly select features that could be predictive for cell type, and that would be something like the intensity in the image data. So I will choose the mean, the min, and the max intensity. Then I will choose the relative movements to other cells as well. In this example, I will use the spot ellipsoid properties. So for example, in this case, the volume. I will add the aspect ratios. So these are the ratios between short and middle and long semi-axis. So that's also indicative for the shape. And I will also add the displacement or the velocity. And then if I have made my choice, I can compute the Umap. This may take a while in this little data set. It goes really quick, but if you have bigger data sets, this at the moment can run quite a while. Then when this is computed, you can see that the Umap output is added as columns to the data table that you can see here. And this you can then again show in the grapher. So I will plot here Umap 1 against Umap 2. It shows the full graph, and I will plot the results. And I get 2 nice clusters that I can identify. And I would like to visualize these now as well. And for this purpose, I select configure text. With the configure text, I can add a tag set. So for example, I add a tag set Umap and I add cluster 1 in purple color and then cluster 2. So for example, take like blue color, apply. So after creating these tag sets, I would select all those points or spots that are in this cluster. And I can select then the tag, so that could be cluster 1. And I can already apply that coloring, so that is then in purple. And then I would select all the spots in cluster 2, can select them, and then, yeah, I could also use the same coloring in other views as well. So for example, here in the BDV, you can see it quite nicely, and it's also possible to show the same, for example, in the Blender view if you have that installed. So this would be this data in the Blender view. You can see quite nicely that there's like two main types of cells in this data set, this upper set of cells and the lower set of cells, which separates quite nicely. We could also show the same coloring, for example, in the Umap. Besides the features on the model graph, it's also possible to compute the features on the branch graph features. So that are, if I go here to the branch per table, that would be the data columns that refer to the branches. And yeah, it's possible to compute the Umap also on the branch level. In the current released version of Mastodon, it's not possible to show them, but I can show a preview because that will be possible in the near future. So that's the graph branch view. And you can show the Umap that is computed on the branch level as well. And you could then select here as well clusters if you want and show them as well. In this release, we decided to rename the function lineage classification to a hierarchical clustering of lineage trees. This function can be used to compare cell developmental patterns and group cells with similar patterns together. We decided to do this renaming because we think that this hierarchical clustering of lineage trees more precisely reflects what the function actually does and how it works. Because if you look in the track scheme, the function would group lineage trees that look similar to the eye to the same group. Another new function that we have added with this release is that you, in case you have multiple similar movies of the same object, you can compute an average hierarchical clustering, including and reflecting all these similar movies. In order to demonstrate this, we are using movies that have been published with the Science article, and the dataset has been published by Patrick Lemaire, and it contains 10 embryos of Phallusia mammillata, embryonic development. All of these 10 videos show the embryos between at least the 64 cell stadium and the 200 cell stadium. So you can see here in this example, this is dataset 8 and this is dataset 6. You can see that they look similar, so this dataset goes until the stadium of 300 cells, and this goes even further until the stadium of 400 cells, but it shows similar development. And yeah, in total, 10 videos exist. Hierarchical clustering of lineages can be used to study the relationship between cell development patterns and cell fate. And it is possible now to increase the accuracy for such studies by including multiple videos of the same organism. The similarity matrix that you can see here, that's just an example similarity matrix that is not visible to the user, but is used. The similarity matrix, which is the basis for the actual hierarchical clustering, is computed for each project individually and is then averaged after it has been computed for each project individually. And with this, it is possible to increase the accuracy for such studies by including multiple videos of the same organism. An important precondition to do so is that the naming is consistent. So you can see here, for example, that the cells have this name A7.0001 star, and so on and so forth. And you can see in this second data set that there's a very similar and consistent naming scheme. So that's an important precondition, because the similarity matrix uses these names, and if the names are different, then they cannot be matched together over the multiple projects that you can use. And a second important precondition is that the cell states that are used for cropping. So in this dialog, you can say, okay, for my analysis, I would like to investigate the cell lineages, for example, between the 64 cell states and the cell state of 200 cell, let's say, and they need to be in both of the. So what you can do, you can add these 10 Mastodon projects of the same or of the similar organism, showing similar development and having consistent naming in them, to this dialog, and then you can set like the crop criterion. So I would like to start at the 64 cell state, and I would like to end at the 200 cell state. And it is important that all these 10 datasets contain these states. So for example, if I would choose, I would like to end at 250 cell state, then the validity check of the parameters would tell me that, okay, in this project, embryo PM7, Phallucius mammillata 7, the maximum number of spots, so cells, so to say, is 217. So I can only, if I want to use all of them, I can, could do until state 210, and then it's valid again. So if I have valid parameters, I can choose the number of clusters. So I choose 12 here, for example. And I can also decide that I write the generated text. So that's the classification that you will see in a second, that you also write that to these other projects. And it's important to note that this will write these tags really to these other projects. So if you don't want your other projects be changed just by this analysis, you should not check this. But if you want to have these average tags in all of your datasets that are included in this analysis, that's possible with this checkbox. Then you can press OK, and it will compute the average classification. And as you can see here, not all of them are annotated with a cell fate, but many of them that fall in the same group, so that show a similar developmental pattern, they also end up as being in the same class of this lineage clustering. Another functionality that we have added with this release is the export of the clustering result to CSV. And the result of this export just looks like this. So you can see here the groups, and you can see the similarity groups. You can see the cell names, and you can see the fates. And this basically shows the same as you can see here. So for example, all these cells that fall in this group 12. So most of the head epidermis cells that you can see here, they are in the same group here. And this is the similarity score. And the lower the similarity score, the more similar the cells are in this cluster. Another example would be all of these anterior, dorsal, and ventral neural plate cells. They fall in group 8, and they have dissimilarity score.